Hello, everyone, and welcome to today's webinar, Overcoming Challenges with AAA Aggregate Analysis using a novel set column to determine critical quality attributes. Today's educational web seminar is presented by LabRoots and brought to you by Phenomenex. To learn more about our sponsor, visit phenomenex.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you experience any technical questions, you may utilize this Q&A box and we will assist you directly. I'd like to now turn the webinar over to our moderator, Dr. Ram Dadapani, Senior Product Manager at Phenomenex. Dr. Dadapani, over to you.
Hello everyone, welcome to this webinar. Today we will talk about how to overcome challenges with AAV aggregate analysis using a novel size exclusion particle. My name is Ram Kumar Dandapani. I'm a Senior Product Manager here at Phenomenex. Joining me is Dr. Shilin Cheung. Dr. Cheung has received her BS and Ph.D. in Bioanalytical Chemistry from University of Toronto. She has developed, qualified, and validated innovative analytical methods in capillary electrophoresis-based microfluidics, automated low-volume UV spectroscopy, and ultra-high performance liquid chromatography assays. Her methods have been applied to manufacturing process development, formulation, stability, drug substance and drug product characterization, and specification testing. At Phenomenex, she's focused on developing fit-for-purpose analytical methods to help advance development in gene therapies. Here is a brief overview of today's presentation. So we will focus on adeno-associated viruses. What are adeno-associated viruses? Why aggregation analysis? And then what are different AAV serotypes? We'll also talk about common challenges associated with AAV analysis. In the next part, we will speak about the optimized solution for AAV analysis, which could reduce sample consumption as well as improve resolution through pore size optimization and hydrophilic chemistry and inertness. We'll also look at how this column is versatile with different mobile phases and detectors. And last but not least, we will also explore batch-to-batch reproducibility. Right after this, I will hand the floor to Dr. Shilin, where she will be exploring method optimization and extended applications of AAVs. AAVs are common vectors for gene therapy. They have wide range of tropism, meaning they have specific action towards specific cells in your body. There are 13 known AAVs that can specifically act on, for example, brain cells. AAV1, AAV2, and AAV9 can actually interact with brain cells. Likewise, there are other AAVs that are target specific to other cells. In terms of mode of action, basically the AAV gets engulfed and then gets lysed. The payload inside the AAV gets into the system. And then because they are able to carry a large amount of gene material, it's very easy for patients to get these to target specific sites, which are very hard to reach with traditional therapeutics. So because of the large load, the patients can also take these drugs less frequently. In addition, the AAVs are also non-pathogenic, because of which there is very less adverse effects. Because of all these reasons, gene therapy is gaining a lot of momentum. And then there are at least 5 known AAVs that are already approved, and a lot of research is going on in clinical trial for further exploration of clinical trials. Now, an important aspect here to actually find out the quality and ascertain the critical quality attributes is the aggregate analysis. So aggregation of AAVs actually has a detrimental effect on the efficacy of the gene therapeutic itself. So it's important to understand the aggregation level as well as identify exactly what is happening so that the efficacy can be improved as well as the recovery of the drug can be improved from a drug manufacturer's perspective. So having that in consideration, size exclusion is a great alternate here to actually look at the aggregate profile. Now let's talk about AAV serotypes. There are 13 known natural AAV serotypes, and as you could see here, the AAVs look icosahedral in shape, and they have a size of 25 nanometer. The capsid ratio of the subunits, it has 60 subunits, and the capsid ratio of the viral protein 1, 2, and 3 are in the ratio of 1:1:10. And they also have a viral genome, which is 4.7 kb with ITR on both the sides. The capsid also has constant regions and variable regions. The variable regions are the one that makes these specific and selective to specific organ tissues. So now that we have understood about different AAV serotypes, let's talk about what are the common challenges that people face when they want to analyze AAVs and then characterize those AAVs. Here are a couple of challenges that people come across. The primary challenge that people come across is the resolution and improvement of data quality. So improving the resolution actually helps estimate the aggregate level as well as accurately integrate, and using multiple detectors, you could identify the molecular weight of that specific aggregate. So it's important to have an optimal column that can improve the resolution and provide a wider range for resolution improvement, thereby improving the data quality. And when we talk to these customers, sample consumption is a big deal for them. The samples are very scarcely available. So if an analytical technique could save sample by utilizing least amount of sample for the analysis, the remaining sample can be utilized for other testing purposes. So it's important to consider those in mind when talking about these analysis. Now, in addition to that, people are also looking for better resolution as well as high throughput methods which can have shorter runtime. Traditional columns as well as traditional techniques like ultracentrifugation might take an hour long, while with optimized column dimension, you could get short runtimes and improve the throughput of your analysis. In addition to that, having high recovery actually will give you the confidence on the analysis and also improve the lifetime of your column. For example, if there are hydrophobic impurities in your sample and then it gets adhered to your column, that might sometimes even reduce the lifetime. So getting high recovery not only improves the data quality, but also it improves the lifetime of your column and method. So there is certainly a need for optimized column for the analysis of AAVs. So having all these in mind, Phenomenex has developed a specialized particle, which is Biosin D6-7. So if you look at the particle, it is a high efficient 3 micron fully porous particle. The large pore size of this particle can accommodate large proteins like AAVs and IgMs, and also it has the diol selectivity, which is highly inert. With the diol selectivity, you're able to get true size exclusion and no interaction with the analyte, so you get highest recovery. In addition, the flow path of the column is inert, which actually improves the recovery and resolution of analyte. We also will demonstrate excellent batch-to-batch reproducibility in our future slides, and we will explore a lot of applications related to this. So now, talking about size exclusion, how big are we talking about, right? So size exclusion is a hydrodynamic volume-based separation. Biozen is a platform where you get that inert path for your analyte to get these separations. DSEC7 is a 700 angstrom pore size particle, while the DSEC2 is a 200 angstrom pore size particle. Both these are diol-based selectivity, so you get no interaction and pure size-based separation. While Biozen DSEC2 is well positioned for MABs analysis and proteins of that size, Biozen DSEC7 is specifically designed for AAVs and IgM sized proteins. So when it comes to analysis of AAVs, one key parameter to consider is sample consumption and also improving data quality. As we know, the traditional technique for these analysis has been AUC, which is an ultracentrifugation-based technique. While this technique was used in the past, there is a lot of movement towards size exclusion because of the robustness of size exclusion, as well as the sample consumption aspect. As you could see here, a general AUC method would consume 400 microliters of sample, while with DSEC7, small sample volume such as 2 microliter is more than sufficient. With traditional SEC as well, with larger ID dimensions, it would utilize 20 microliters. And then in terms of runtime, when we compare AUC and traditional techniques, DSEC7 can provide you short runtime as short as 7 minutes. As you could see here, the AAV profile of aggregates for AAV2, we have injected several sample volumes of 0.5, 1, and 2 microliters. Even at the smallest injection volume of 0.5 microliters, you're able to accurately determine the aggregate levels of the higher molecular weight segment. So this is possible because of the inert surface, which provides you higher recovery so that you can go to lower level injection volume and still recover those analytes out of the column. And in addition, with the optimal dimensions, you're able to reduce the runtime as short as 7 minutes. So if you'd like to further reduce the sample volume, you could also go to a smaller ID column. As we show in the example here, the top chromatogram is the high level where we are injecting 0.5 microliter on a 4.6 mm ID column and a 0.1 microliter on a 2.1 mm ID column. A zoomed version of it is shown here in the bottom figure. As you could see here, the high molecular weight aggregates are clearly separated even with a 1 microliter injection. So this is possible because of the inert surface chemistry of the particle along with the inert hardware. So now let's look at how the pore size actually affects the resolution of AAVs. Here we have two specific examples. We have AAV3 and AAV8. What we have done here is we have explored multiple pore size particles that's commercially available. And as you could see here, when we start with a 200 angstrom, there is not much separation of AAV monomer from its aggregate. And then once we go to 400 or 450 angstrom particle size, you're able to see resolution. And with the optimized pore size with the 700 angstrom, you're able to enhance the resolution of aggregates from the monomer. While developing Biozen D6-7, we explored multiple pore sizes and optimized our separation to get maximum resolution of aggregates from the monomer. As you could see in the bottom, there is resolution improvement that you could see from a 450 angstrom to a 700 angstrom particle size. So two main factors that are improving the resolution here. One is the optimized pore size, and the other one is basically the chemistry of the particle, which is hydrophilic, which is improving the resolution as well as recovery. So here is a zoom into the chromatograms of AAV8 and AAV3. As you could see here, with a 700 angstrom Biozen D6-7, the details of the aggregate can be explored. Especially if I couple this to a mass detector and I wanted to identify the molecular weight of this specific fragment, it's way easy because you're getting way more details of the analyte and more separation. That's possible because of the optimized pore size. Due to the optimized 700 angstrom pore size, you have a larger range for improving resolution of the higher molecular weight components. And then the same is evident with other serotypes as well. On the right side, you could also see that with AAV3, an enhanced resolution on Biozen D6-7 with a 700 angstrom particle. So here is another example where the inertness helps you achieve better lifetime as well as recovery. As you could see here, we have the analysis of IgM on a 450 angstrom particle as well as on Biozen D6-7. As you could see here, the later eluting sticky impurity, hydrophobic impurity, gets better recovery on Biozen D6-7 within 7 minutes. Basically, this would have otherwise stuck to the column and it can actually accumulate over time and it could actually deteriorate the lifetime of the column. With Biozen D6-7, because of the diol chemistry, which makes it highly hydrophilic, you're able to get pure size exclusion based separation. So even the most sticky sample doesn't stick inside and you get good recovery and you could extend the lifetime of your column. So now let's focus on an example here. Here we are showing a comparison of traditional particle with a 750 angstrom pore size versus Biozen D6-7, which is a 700 angstrom pore size. So here, if you look at the profile of the peaks here, two things that can be paid attention to are the peak apex. If you look at the peak apex, Biozen D6-7 shows you higher efficiency compared to the traditional selectivity. And then when you look at the resolution as well, there is an improved resolution for the aggregates. So the reason for the improvement in peak efficiency comes from three different places, right? So the particle is optimized with high efficiency particle size. Biozen D6-7 is a 3 micron high efficient particle size with a very tight pore size distribution, because of which you get high efficiency. And even better, it's packed with higher efficiency. So all these three components together contributes to better peak response and recovery. And then in addition to that, you also have the hydrophilic surface, which actually promotes true size exclusion based separation, and the inert path for the analyte makes it recover the sample. So combining all these benefits, you're able to see better separation as well as better recovery on Biozen D6-7. Now, when we want to determine the molecular weights, it's traditional to use Mal's detector for this analysis. Mal's detector is very popular and it's also very helpful to determine the molecular weight because you don't need a calibration curve to run for estimating the molecular mass. And also, another aspect to consider when you're using MALS detector is that you have to allow enough time for the detector to stabilize in order to get stable baseline. With BIOS and DSEC7, because of the modern particle technology and the inert surface, as a plugin, the column gets equilibrated as well as your detector gets equilibrated. We have a couple of tech notes that talks about the separation as well as use of multi-detectors using BIOS and DSEC7 that can be downloaded free from Phenomenex website. What we have seen in our experiment is the column just as a plugin would stabilize the detector without much increase in the baseline. In addition to that, it's also very important to have a column that's versatile with multiple mobile phase. Because of the surface chemistry, the column is already inert and it's hydrophilic. So it's compatible and flexible with multiple mobile phases that are commonly used. People that are analyzing AAVs, they wanted to look for a column that they could use for a platform method. So if there is one specific method or one specific mobile phase parameter, they could run multiple serotypes using that. That is a throughput improvement for the lab. So as you could see here, we have taken AAV5 and we have ran it across multiple mobile phases. What we have noticed is there is minimal change in the aggregate percentage or resolution with different mobile phase conditions. If you look at the 10% ethanol, there might be a slightly broader peak, but when you have really sticky sample and you want to remove those hydrophobic impurities further from the column, 5 to 10% ethanol would help you with the size exclusion. So the takeaway here is the column is compatible with multiple mobile phase conditions, so you can confidently use it for your platform method across various mobile phases. And here is an example of the robust nature of this column and also the manufacturing process that we wanted to demonstrate. We want to make sure the column is useful throughout the life cycle of your method. What we are demonstrating here is 4 different batches of the BIOS and DSEC7 media that we packed, and you could see that the retention as well as profile are spot on, and there is minimal variation between batch to batch. Basically, this gives you the confidence to make a robust method that can go through the life cycle of your method. We have Dr. Shilin Cheung here to talk about method optimization for AAVs with BIOS and DSEC7. Over to you, Dr. Cheung.
So in my section, I'm going to talk about the experience I gathered from developing size exclusion chromatographic methods for AAVs utilizing the DSEC7 column. I'll be going over the LC system requirements, typical operating pressures to be expected, the type of buffers that I've experimented with, as well as sample preparation and the flow rates, controls that I've used, as well as I'll be sharing data on quantitative methods where I look into AAV serotypes, empty versus full, and also into AAV stabilities. Lastly, I'll be introducing the combination of utilizing our DSEC2 in series with our DSEC7 column to look at a wider range of hydrodynamically different species in my AAV sample. So firstly, about the LC system requirements and the operating pressure. Our BIOS in 3 micron DSEC7 column, the 30 centimeter long format, was designed to operate at approximately 150 bars. This translates to 2,200 PSI. And if it was 150 millimeter or a 15 centimeter format, it would be approximately 80 bars, so half of the 30 centimeter. This means that you don't necessarily need to run this column utilizing an UHPLC system but you can also use just an HPLC system instead. Now, if you wish to have higher resolution separations, I would recommend you minimizing the volume, meaning cutting all the tubings, minimizing the length there, to avoid sample dispersion. So comments for the buffer, sample preparation, and flow rate. So this column actually utilizes the same chemistry as our DSEC2. As such, any typical site exclusion chromatographic buffers can be used. And in particular, if your AAV buffer is a typical of PBS, with some pluronics, you can actually utilize it on this column as well. I've also used a low percentage of SDS in certain cases, and I haven't noticed any adverse effects. The pH stability of this column is from 2.5 to 7.5, and it can tolerate a wide range of ionic strength. So within this salt mix, typically I would have about 10 to 50 millimolar of a buffer, sometimes a sodium phosphate, combined with approximately 150 millimolar of sodium chloride. For some AAVs that I've worked with, I found that it is critical to go above 200 millimolar in sodium chloride content for it to elute off the column correctly. With regard to the sample preparation, it really depends on how your sample is when you received it. If it was freshly prepared and you don't notice any particulates in there, and you're not worried about it clogging up your LC system, you could inject it straight. Alternatively, if you're worried about it clogging up your system, you could choose to centrifuge it or apply a 0.2 micron filter to it as well. For a lot of these AAV samples, because your running buffer is actually your formulation buffer, you could also create dilutions of these samples and utilize that to actually verify the recovery, to look at the stability in a number of different buffers, and also even to look at the stability of your sample over time. Now, note that the LC itself, the column itself with the detector, actually has really, really good precision. Meaning, typically, if you inject something, it gets to the system, you only have maybe an RSD of 2%. So you can actually utilize the accuracy and precision of the LC to help you establish a very robust and reproducible sample preparation method. For high resolution separations, I would suggest using the 30 centimeter long column at a flow rate of 0.25 mils per minute, where the AAV monomer elutes at 12 minutes. For fast separation, I would suggest utilizing the 15 centimeter long column and running at 0.5 mils per minute, where the AAV monomer would elute at 3 minutes. So with respect to controls, during the method development, often you want to check your column, often you want to check your LC system, as well as your detectors. And because AAV samples tend to be quite expensive and limited in volumes, a couple of standards that I found to be, or controls that I found to be quite useful are the following. The first is the Faro goblet, which is approximately 670 kilodaltons. Alternatively, you can use something that's quite sticky, like BSA. You can utilize antibodies such as IgM, which is approximately 900 to about 1,000 kilodaltons, IgG if you wanted, or uridine as a marker to mark the total inclusion volume as well. Now, for myself, when I develop these methods and create the sequence of injection, often I would use these controls to inject right in the beginning after I've set up my system and make sure that these injections of these controls actually produce enough, high enough reproducibility in both peak area and retention time before I start injecting and analyzing my samples. In addition, after I do that initial check in the beginning and start running my samples, I would continuously bracket my samples with these controls. Now, the benefit of that is at the end of your run, before you analyze your samples, you would go in and try to overlay all these controls. And what's really good is if you just overlay them, you can actually visually check how good is the reproducibility in peak area and retention time. So what you see here at the bottom is an example of overlaying 2 injections of this control in one of my studies, AAV studies, where they're separated by 13 injections apart of AAVs. And if you look at carefully, you'll see that actually the first injection was actually in the blue trace. And because of the reproducibility of the column and nothing getting stuck, we actually see on the injection 14, the red trace to be exactly overlaying with the blue trace. Now this, without even data processing, I can already tell you that I have confidence that my samples, which sandwich between these 2 injections, are likely to be, behaving as expected. And therefore, then I would go in and start analyzing my AAV samples. One more thing that often gets overlooked is do inject the formulation buffer blank. This often will help you to identify peaks of interest. In addition, you could also look for the total inclusion volume. Here, you'll see the uridine marker actually marks that as well in the UV-280. Now I'm going to jump in to show you guys some data. So when we talk about AAV quantitative methods, one of the questions that often come to mind is how good am I at recovering my monomer from this column? How good am I at recovering the aggregates or the impurities of this column? Now, one can resort to injecting the sample just utilizing a union, or we can dilute the samples down and take a look at across the actual analytical dynamic range, how well it performs. Alternatively, I can take other columns, whether it could be an ion exchange column or another markedly available site exclusion column, and compare the peak areas. Because at the end of the day, if it's in a similar buffer matrix, the largest peak area means you're recovering the most off your column. So here, on the graph on the left, you'll see a comparison of one of the red peak is from a commercially available column on the market versus our BioZen D67. What you see here is that on our BioSec D7, you actually recover the AAV with a much more Gaussian looking peak shape, as well as the peak height is also higher. So meaning, even if I don't have to do the math here, I have a much more narrow peak, much more taller peak, which actually does to my advantage when it comes to quantifying this peak in terms of its peak area. On the right over here, what you see is a bar graph doing this comparative study across a number of different AAV serotypes. So here in the blue is the data from a BioZen D67. The red and the gray are from 2 other commercially available columns that are on the market. The peak area is shown on the left here. This is the total peak area of the AAV monomer plus any impurities or aggregates that you have within your sample. The bottom here again is the AAV serotype. When you look across this data, what you see is our BioZen D67 actually recovers the same peak area or slightly a little bit more in peak area than all the other commercially available columns out there. Now, knowing that our D67 seems to recover both the monomer and the aggregate really well, I decided to develop a quantitative method for this. So in this example, the sample prep is the following. I purchased the sample commercially from a vendor, and what I did was I decided to take a look at the recoveries as well as the linearity across a 10-fold dynamic range. So from one single sample, I separately created 5 different samples from it. The first one was a 1 to 10 dilution in the formulation buffer, which is also my size exclusion, the sec running buffer. And then to the most diluted sample, which is basically a 1 in 100. After I diluted it down, mixed it really well, I would load it into my sample vials. And what I was really interested in is if I was to really develop a very robust method, I would need it to work on more than one D6 column. So this study, I actually got 3 different columns from 3 separately manufactured columns. So these were like both the chemistry as well as the filling, everything was done separately. So there's a completely 3 different lots. And I ran it. So what I did was each column, every single sample, I injected 3 times. Here on the right on the top is a plot of every single concentration, every single sample that I had, injected 3 times across 3 columns. This translates to a graph of 45 separately plotted points on it. So every X is 1 data value obtained. If you want to see what the chromatogram itself looks like, you could look down here. This is the AAV monomer of interest. This is the aggregates and the impurities that are looped towards the left of the AAV monomer. When I zoom in, I can see a concentration dependence of the signal of both the aggregates as well as the monomer itself. So what can you tell from this graph up here? Number one, what I see is that there's really high linearity, meaning for everything that I inject, I get a corresponding peak area response from it. And the R-squared is basically 0.999. And if you look at all the separate points, you can see there's very, very little difference in the actual values. And this also gives me confidence that across this dynamic range, I'm having reproducible or almost 100% recovery of this monomer. Now, you may ask me, how about the aggregates? So if I was to look at every single point across this data for n equals to 45, and I look at the max and the min percentage of monomer that has been determined, across those 45 points, they only range between 92 and 94%, meaning plus or minus 1 from the average of 93. So what this implies to me is, 1, I can reproducibly, recover the monomer, and as well as reproducibly recover all of these impurities and aggregates across a large dynamic range, which gives me a lot of confidence that whatever I inject into this column, it is indeed coming out. The second study that I did was, because we have such good reproducibility in retention time, I decided to see if I can see any type of difference amongst the various serotypes of AAVs. So what you see here in these data points here, first one is actually of AAV1, then I have AAV2, AAV3, 5, 6, 8, 9, and 10. For reproducibility and assessment, what I also did was I took a look at 2 different injection volumes to make sure that no matter how much I was injecting here, I was getting the same percentage of the AAV monomer being recovered. And you can see here, it's highly reproducible, no matter whether I was injecting 5 or 10 microliters. Then if I take a look at the retention time and try to explore whether I can see any differences in the serotypes, this table down here lists them in order from the quickest eluting to the slowest eluting one. And from observation, what you will see is AAV1 and AAV5 shows the greatest difference in the retention time. And if I go and talk to AAV manufacturers or people who actually study these things, they would tell me that AAV1 and 5 are indeed serotypically the most different. If I look at AAV1 and 6, I get a reproducible difference in the retention time as well. And when I look at their amino acid sequence, I realize they only differ by 6 out of 736 amino acids. Now, if you ask me, if you give me a mix of everything, would I be able to separate them all and quantitate? I would say no, because based on the retention time, all I can do is do some type of identification. But nonetheless, I do see a retention time shift of this peak dependent on the serotype. Now, with this result, another thing that was very interesting was then I asked myself, is there a difference that I could see in the empty versus a full? So in the bottom graph down here, you see is an overlay of the blue trace, which is the AAV9 full AAV versus the AAV9 empty, which is shown in the red. If I run them day to day and verify and make sure there's really a difference, what I see is for at least two additional serotypes, the empty one always alludes to the left of the full AAV. Now, if you look very carefully at these two differences, probably only maybe like a 3 or 4 second difference. Again, if you ask me, if I have a mixture of the two, will I be able to separately quantitate them? I would say no. But if you were to give me a sample and ask me to identify whether it was AAV9 empty or an AAV full, I would be able to tell you, because all I have to do is take a look at the retention time as well as the impurity profile, and I would be able to identify which one is which. So the last data I want to show is AAV stability. So with this particular AAV, what I did was I decided to expose it to an increasing percentage of organics. So here on this graph on the left, what you see here is the AAV monomer by itself. The blue is this control sample, which is, pure AAV8 in PBS with 0.001% pluronics added inside, F68. As I add in a little bit more, 20%, 30% of organic in there, what happens is the AAV monomer starts decreasing. And then these impurities, or so-called aggregates, impurities, start increasing correspondingly. So what's interesting from this is one, the magnitude of these impurities and aggregates are dependent on the percentage of organics that I put in. And secondly, is that these impurities or aggregates that are generated are well separated from my AAV monomer. Now, if you think about what it implies is that the column actually did its job. It allowed you to separate the forms, the forms that are inside the sample really, really well. So now you can bring your detectors in and start to identify what these things are. So here on the left, this is actually a UV280. You can start looking at the FLD response. And what you can also do is start looking in the ratios of 260, 280 of the separate peaks. For example, this mean peak always stays at 1.3, which is as expected of a full AAV. And then these ones up here, this first one actually has an A260 to A280 of 1.46. But if you look at this one on the far end, it actually has 1.8. And it's only the separation that allows you to start bringing in detectors and really allowing you to identify what these things possibly could be. So the last set of data I want to show is the enhanced utility of utilizing two of our products that are utilizing the same chemistry together. So what I've done here is I've combined our DSEC2, which is 200 angstrom size exclusion column, with our DSEC7, which is our 700 angstrom size exclusion column. And the way I've combined them is after your injector, it would first go into our DSEC2 column, then you would combine it to our DSEC7, and then you hook it up to your detector. Now, under this configuration, if you choose to use a 15 centimeter long column, you would expect a pressure approximately 140 bars. The effect of adding these two together, I'll show you over here. If you just have the DSEC7 column by itself, what you'll see is the lower molecular weight range, which is less than around 100 kilodalton range, they will look kind of all bunched up together. It could be very difficult for quantitation or identification of these species. Now, if you hook it up with our DSEC2, what you'll see is this range now gets magnified and basically spread out because of the DSEC2 to look like this. Now, if you imagine, if you have your AAV8, right, eluting around here, then you can look at your impurities and the aggregates over here, as well as the lower molecular weights or the hydrodynamically smaller species on this side. So within the same run, one run of basically 15 minutes, you'll have this enhanced capacity to separate both the large species as well as the smaller species. So with this, thank you for your time. And I'll hand it back to Ram.
Thank you very much, Dr. Shilin. That was very interesting study and a lot of information about method optimization. So this brings us to conclusion. Here are the key learnings. AAVs are attractive vectors for gene therapy, and then there are certainly some challenges which DSEC7 addresses. As a holistic solution, DSEC7 provides you reduced sample consumption, shorter runtime, utilizing improved resolution through the pore size optimization, and with hydrophilic chemistry and inert path. Biogen DSEC7 is also versatile with multiple mobile phases and detectors. We have tons of resources available on Phenomenex.com for free for you to download. In addition, Dr. Shilin also explored a lot of method optimization tips and tricks with you, and also discussed about enhanced application by combining DSEC2 and DSEC7. Now that we discussed AAV aggregate analysis, we can also look at complementary applications like intact VP analysis using Biogen Intact XBC8, as well as the peptide mapping using Biogen XBC18 in a 1.7 or a 2.6 micron particle size. Thank you very much for listening to this presentation. Now I'll open the floor for questions related to this presentation. I believe we have a couple of questions coming in. For people who have sent questions, thank you very much. We still have time to take a couple of questions. Feel free to send additional questions through the comment box. We'll answer a couple of questions during the time. And if we are not able to answer it within this time, we will send you an email reply with an answer. So here are a couple of questions popping up. The first question I have is, what is a good starting point mobile phase you would suggest for AAV aggregate analysis?
Yes, Ram. So the starting point, I would say, is basically the formulation buffer. Whatever it's running, and typically what I've seen the most is approximately like 1 times PBS with about 0.001% Pleuronix F68 inside. There's no problem with starting with this composition as your running buffer. Sometimes if you do see that your AAV monomer peak, which is basically your main component, is not coming off, I would recommend you quickly pushing it, the salt content, your sodium chloride, going up to beyond 200. So something like maybe around 300 millimolar to see if you get better recovery of your column.
Excellent. We have another question. They're asking if we have any tips and tricks to improve the sample preparation.
Yeah, so with regards to AAVs, in my experience, these are actually very sturdy biologics. Typically, I found them to be quite more robust than some of the proteins that I've worked with in the past. So typically when I get them, right, sometimes if they are frozen, I would thaw them. Most of them, I don't actually think there are any aggregates or anything that would actually clog my LC system. So in some cases where I'm really looking to see what's in there, and I don't want to in any way, centrifuge and cause things to precipitate out, or apply a 0.2 micron filter and get things stuck in the filter, then I would just try to inject straight, at least a small volume of it, monitor the pressure on the LC system. If it doesn't clog, then go ahead. If you do see or suspect that there could be something that could be clogging up your system, right? This is, again, particular to HPLC or UHPLC, where the tubings are very, very narrow. then automatically you would at least try to centrifuge it, allow at least about 10,000 RCF, or apply, if you have sufficient volume, to filter it through a 0.2 micron filter, and then analyzing from there. So yeah, sure.
All right, here is another question. Based on your experience, what type of performance can we expect from various batches of this column?
Yep. So right now, based on my experience of running at least 3 different manufactured columns, so meaning different lots, different batches, what I've seen is the recovery, there is no difference. We always recover 100% of this AAV. Now, with regards to the retention time, there might be a very, very minor shift from lot to lot, but the shift is actually, probably talking about, 2 or 3 seconds across a 3-minute elution of the AAV. So there is absolutely no problem with identifying which one is your AAV monomer versus any impurity or aggregate.
Excellent. Thank you so much. That's very helpful. I have another question from a customer. He's asking, currently I'm using ultracentrifugation technique for AAV size variant analysis. I'm looking to transition to size exclusion. What are the things to expect? And then what are good starting points for me?
Yeah, so whenever you're looking to transfer a method to look at the different size variants, one thing you always have to know is kind of the size limit, right? And it's all stemming from all the way to sample preparation to the analysis itself. So typically for HPLC, if you run anything on HPLC or UHPLC, there's like tubings and injectors. They all have very, very narrow IDs. So if you talk about, if you're interested in looking at things that are really, really large, like micron size or really, really large aggregates, right? You will not be able to see that on the size exclusion chromatographic column or any type of LC column if you're injecting into actual LC system. I would say, right, based on what I'm looking at, the largest sizes I've seen coming off our D6-7 column, it hovers around about 10. I've seen things as large as 10 megadalton popping off. Again, for these really large biomolecules, right, you start looking at conformation, right, whether this 10 megadalton species is spherical in nature or linearized in nature. You start seeing those effects as well. So I would say size exclusion with a SEC column, D6-7 in particular, you get really good accuracy and precision for looking at specifically at the monomer. And these, maybe dimers, trimers, species, you'll get really, really good recovery and quantitation, much better than any other size exclusion methods, size type based methods that you have out there. So that's what I would say to expect. And again, in my experience, if your monomer, if you run it on the SEC and your monomer goes somewhere, right, or you start seeing a decrease of it and you don't see it popping off the column, then I would suggest you bring in another technique and look for something that's really, really big. But that ability to really accurately and precise wise, robustly look at that monomer is really the key thing that we can bring in with this D6-7 column.
Excellent. Thank you so much, Dr. Shilin. This is very helpful. In addition to that, I would also say you can save on the sample, right? So you may not need that much of sample. You don't need a 400 microliter or 500 microliter sample. It's just few microliters, 3 to 5 microliter sample injection. And also your run time is going to be really short with your size exclusion, as short as 5 minutes to 7 minutes. Excellent. So we'll take one last question. This question is, are we in a fear to shed the AAV if we run at higher flow rate?
Yeah, this is a great question. So these high flow rates, if you look at what is commercially, out there, some 7.8 mm columns actually run at 1 mil per minute or even higher. So when I'm doing method development, I often look at the percentage monomer or the aggregates that are recovered at different flow rates. As of now, I would say I haven't seen any effects on the AAV or its associated impurities up to 0.5 mils per minute. Beyond that, if you go much, much higher and start squeezing these samples through narrow capillaries, I'm not so sure. But if you were to run a 4.6 by 15 or 30 centimeter, a 0.5 mils per minute should not be creating or reducing any other species that's not already inside your sample.
Excellent. Thank you very much. Thanks to the audience for the questions. And thank you so much for listening to this webinar. We'll come back with another webinar soon. Until then, thank you very much.
Thank you again, Dr. Dandapani and Dr. Cheung, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Phenomenex, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period, they will be addressed by our speakers via the contact information you provided at the time of registration. Today's webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Thank you for joining us. Bye-bye.